Well, thank you for inviting me to present the results of NSABP-B-47, presenting on the as a representative from all the investigators from NSABP and NRG. So you've read the title already for me. These are my other investigators on this study. Here are potential conflicts of interest for our physicians, most of which are conducting the studies themselves. So, although this may not be a medical group, you're probably quite aware of the 2005 ASCO presentation by Dr. Ramond, in which we were presented with the stunning results of the effect of trastuzumab used in an adjuvant fashion on HER2-positive breast cancer. This was for patients who were IHC 3+ or had a FISH score of greater than or equal to 2.0. And you can see these curves separating dramatically for disease-free survival and unheard of p-values showing this to be a very dramatic effect. A great time for oncology. A review of those studies, B31 and N9831, it was a combined analysis. The chemotherapy was AC followed by paclitaxel with or without 1 year of trastuzumab. This was in HER2 high-risk patients. All were node positive at the time. And importantly, to focus on, the initial study entry criteria included the FISH test being greater than or equal to 2.0 and an IHC of 3+. This testing was performed at the local laboratory where the patient was being treated. The plan was for a tissue specimen to also be sent to the NSABP for later testing. The testing by the NSABP showed that actually 10% of the patients, 9.7% on central testing, were found not to be IHC 3+ and FISH positive. They were found to be what's called HER2-low, neither one of those qualifying to be HER2 positive. And lo and behold, when the analysis was performed looking at the benefit of using trastuzumab in these 174 patients considered to be HER2-low, it was found that the result was essentially identical, that the hazard ratio was less than 0.5, the benefit was equal, and statistically testing this, there was no interaction between level of FISH or IHC testing, and this was quite bewildering. The hypothesis had been that it would be only the HER2 amplified patients who would benefit from this. So a lot of testing was done. These 174 patients had their samples sent to many different laboratories to confirm that they really were HER2-low, and that all showed that they were HER2-low. They were not amplified in these specimens that have been submitted to the NSABP. And because of that, the NSABP and the NCI initiated a trial, and actually this is a confirmation slide that the N9831, the other companion study, also had 100 patients, 103 patients, who turned out to be on central testing. Again, not the local lab, but central testing were HER2-low. And they had the same benefit. Their FISH ratio was also 0.5. Because patients who are IHC 1+ and 2+, non-amplified, are 3 times as common as the HER2 amplified patients, the enormous potential benefit, the NCI and the NSABP initiated the NSABP study B-47, which was a randomized trial looking at patients who were IHC 1 and 2+ and FISH less than 2.0, and were randomized to chemotherapy with or without trastuzumab. This is the stratification variables in that study. They were randomized to chemotherapy of physician's choice. There were 2 potential chemotherapies, AC followed by Taxol, or docetaxel and Cytoxan, referred to as TC, with or without 1 year of trastuzumab. Otherwise, the therapies were the same. The study population was very large. To answer this question, there were 3,270 patients randomized. There was full follow-up on these patients other than 1.9%. The accrual took 50 months, and the median follow-up at the time of the presentation is 46 months. So that's why we did this study. But here is the result. This is the invasive disease-free survival of the two arms of the study with the hazard ratio of 0.9. We could magnify this slide. You still couldn't see a difference between those two arms whatsoever. The event rate was essentially equal. You can see 134 and 130. The patients actually did pretty well. These were high-risk patients. 5-year event-free survival was 89+%. There was no difference. And this is the forest plot for the stratification variables, which showed no hint of efficacy in any of the stratification variables. You can see that the IHC 1+ and 2+ really show no difference. There is no trend for the IHC 2+ patients to do better. If anything, their dot is on the other side of the hazard ratio of 1. There was no difference between estrogen receptor status and or intended chemotherapy. So the conclusions was that the primary objective of improving the IDFS was not met. None of the secondary endpoints were met. There were no trends for efficacy seen. No difference in outcomes for IHC 1+ and for 2+. Severe toxicities were infrequent. There were no new safety signals were seen. Patients did quite well in this study. Actually, the survival, 5-year estimated survival is 95% for both arms. So the retrospective outcome differences between local tested HER2 positive and central tested HER2-low patients identified in 2 major trials are not readily explained. There's no benefit with trastuzumab therapy in patients with FISH ratios less than 2.0 and IHC staining of 1 and 2+.